Today's lesson is on an overview of my favorite biological material, RNA. And as you've probably seen in many of my videos, I love the central dogma of biology, which states that our DNA goes through a process of transcription to create mRNA, or RNA, that then goes through a process called translation to produce peptides, or proteins. But RNA is quite special because there's actually several different types. And before we break down those different types, I want to actually show you what a molecule of RNA looks like. So there's three different components that make an RNA nucleotide, or a building block of RNA. The first is a phosphate group, shown here in blue. The second is a ribose sugar, shown here in green. Now, if you think of DNA, it's actually a deoxyribose sugar, so it's a little bit different. And then a nitrogenous base, which is shown here in red. And this nitrogenous base can adjust. There can be four different types. There can be guanine, or abbreviated as G, cytosine, or abbreviated as C, adenine, abbreviated as A, or uracil, abbreviated as U. And these different nucleotides come together in 100 to thousands of different strings in order to create the building blocks for various proteins that we need inside of our body. Now, when we actually talk about building that protein, that's when we need the three types of RNA. And those three types are mRNA, or messenger RNA, tRNA, or transfer RNA, or rRNA, or ribosomal RNA. And they all work together because we love collaboration. And they work together in order to make that protein. Now, the first component we need is the mRNA, which is going to actually be the instructions to build the protein. And that comes directly from our DNA through the process of transcription. So that's the protein code. Next, we need the amino acids, which are actually the building blocks of the protein. And those are carried to the code through the RNA tRNA. And then lastly, we need something to actually piece those amino acids together. And that's where rRNA comes into. It's a component of ribosomes mixed with various other proteins that when we combine all three of these different types of RNA, we allow for the ribosome to bind onto the mRNA, recruiting the tRNA that's going to drop each amino acid that it's reading on the code in order to build the protein of interest. And every protein has a different code. And we can think about this like we're building a house. Our mRNA are the blueprints for the house. The rRNA are the actual workers. And the tRNA are the raw materials that we need. All of them come together, and at the end, we have a final functional product. And a lot of people ask, why is mRNA so unstable? Or why don't we need it all the time? Well, that's because only a subset of proteins are actually made at once in our bodies. We don't need all proteins to be active. When we're sleeping, we need to have a lot of our protein synthesis shut off, or maybe after we eat a meal, a lot of proteins are actually being made because we're digesting those amino acids. So a subset is active, but also a subset is inactive. And a way we can regulate that is through mRNA. Having more mRNA, we can create more proteins, and then fine-tuning it off. We don't need to have all those proteins activated at the same time. So now that you have this overview of RNA, I look forward to our next lesson where we're going to talk about the actual building blocks, amino acids. So stay tuned next week and make sure you subscribe so you can stay up to date as these new episodes are released.